So in this PowerPoint, we are going to focus on the plasma membrane and how molecules are going to be moving across the plasma membrane. The first thing that you need to notice about the plasma membrane is that it is selectively permeable or semi-permeable, which implies that some molecules can pass through the plasma membrane while others their movement is limited. When we talk about the movement of molecules across plasma membrane, there are two main techniques or mechanisms that moves molecules across. One situation is when there is no energy required, and we use the word passive processes or passive transport. And if we require energy to move the molecules, we refer to that as active processes. For this PowerPoint, our focus is going to be on the first group, or passive transport. Passive transport has three main categories known as simple diffusion, carrier or channel mediated facilitated diffusion, and osmosis. So let's start talking about the concept of diffusion. When we talk about diffusion, we actually require, or I should say, for the process of diffusion to happen, we need something known as concentration gradient, which basically means that you need to have a high concentration in one area and low concentration in another area. When molecules are going to start to move from the higher concentration to a lower concentration, that is referred to as the process of diffusion. This process does not require any energy input from the organism, and hence it's referred to as passive process. However, you do need to know that even though this is a passive process, it still utilizes a movement of molecules called Brownian motion, which simply means when one molecule hits another molecule, they move in opposite direction. So if you have a high concentration of molecules in one area, these molecules are going to hit each other more rapidly, and therefore they're going to move in opposite direction, and they're going to continue to spread from one location to another. That is the force that drives the process of diffusion. The process of diffusion stops when you have reached equilibrium, which means equal distribution of molecules within your environment or within your solution. There are two main factors that can influence the speed of diffusion. The first is the size of the molecule. The larger the molecule is, or the heavier the molecule is, or the higher the molecular mass of that molecule is, the slower that molecule will move. If the molecule is relatively small and have a small molecular mass, the diffusion process takes it's much faster than if the molecule is larger in size. The second factor that affects diffusion is temperature. As the temperature increases, the Brownian movement, which is the driving force for diffusion, increases, so molecules are going to hit each other at a more rapid speed, and therefore diffusion process also speeds up. So increase in temperature and also decrease in molecular mass both increases the diffusion, and if you have lower temperature, you have larger molecules, both those conditions decrease the rate of diffusion. Here is a scenario where we have basically described the process of diffusion in the sense of diagrams. So we have your dye pellet in the bottom of the beaker with basically high concentration at the bottom and almost no concentration in the surrounding environment. As time passes by, the molecules of dye start to be distributed throughout the solution equally until the molecules have reached diffusion. So movement of molecules from a high concentration throughout the solution toward the lower concentration. Do keep in mind that the plasma membrane allows for only certain molecules to be passed through because your membrane is selectively permeable and therefore does not allow all the molecules to diffuse, even though they might have concentration gradients on either side of the membrane. Again, Considering the plasma membrane is a selective membrane, do keep in mind that if this plasma membrane has been damaged in an instance, in cases including burns, what you lose is the concentration gradients that exist on either side of the membrane, and loss of these concentration gradients takes away from the activity and ability of the cell to maintain its homeostasis, and that leads to cell death. An example of that is a concentration gradient that exists for sodium and potassium on either side of the membrane. So if you take a look at a cell, and let's say this is your cell, you have your sodium at high concentration outside the cell, and you have low concentration of sodium inside the cell, And if you look at potassium, you will have high concentration of potassium inside the cell, and that means low concentration of potassium, low concentration of potassium outside the cell. This is the gradient that exists because of the plasma membrane. If this plasma membrane is damaged, this concentration gradient basically is removed, and as a result, cell will not be able to survive. Now, talking about the different types of diffusion, we start with simple diffusion. Simple diffusion allows a small molecules or molecules that are lipid soluble or lack any electrical charges to easily pass through the phospholipid bilayer. So if we take a look at the picture we have here, we have examples of the molecules that do pass through the plasma membrane without the help of any other molecule. They are simply passing through the phospholipid bilayer. Examples include oxygen and carbon dioxide. These are small gases that basically have no charge and can easily pass through. Benzene, which is a hydrophobic molecule, which means they like to interact with lipids, so we refer to them as lipophilic, so they can also pass through. A small polar molecule, such as water and ethanol, can easily pass through. Even though they have charges, they're considered to be polar, and they do have charges. Their size is small enough that they can still pass through the plasma membrane. However, as you notice that molecules that are large and charged, such as glucose, or ions that are still charged and considered to be polar, such as sodium, chloride, hydrogen, and calcium, are unable to pass through without the help of other components of the plasma membrane. So they cannot use simple diffusion. Just a quick correction in the PowerPoint. When it says lipophilic, the lipophilic means fat-loving, which also means hydrophobic, not hydrophilic. So this vocabulary needs to be changed to hydrophobic. Now, let's say you still want to use the diffusion. However, you're not going to pass the molecule directly through the plasma membrane. Rather, you are going to use the integral proteins that are built in to pass the molecules through. One of these mechanisms is carrier-mediated facilitated diffusion. You can see on the lower picture. What you see here is basically substances that are planning to come into the cell or out of the cell, bind to the protein. Protein changes its shape and allow the solute molecules or the molecules to come through. So you have high concentration on this side, low concentration on this side, and carrier proteins are facilitating or helping the molecules diffuse through the plasma membrane. So the word facilitated implies the use of integral protein for the movement or diffusion of molecules. The second scenario we have, or the second facilitated diffusion technique we have, is using protein known as channels or channel protein. So this technique, a full name of it would be channel-mediated facilitated diffusion, and it basically uses molecules and allow the molecules to easily pass through the plasma membrane using the channel protein. Something you do need to know about channel proteins is that there are 3 different types of channel proteins, and they're basically right here. One is leakage. Leakage is basically implies that these channels are always open and allow the molecules to diffuse in or out of the cell based on their concentration gradient. The other type that we have are considered to be gated, which is broken down to ones that are activated by chemicals or electrical signals. So these gated channels, if they are sensitive to electrical signals, we refer to them as voltage gated channels or VGCs. And if they're chemical, we just refer to them as chemically sensitive gated channels. These are extremely important for the functionality of your skeletal muscle, your nervous system, your heart, and other components of your body. Now, if we want to diffuse water, because water is such an essential component of your body, we rely on it. Diffusion of water has its own vocabulary, and that is osmosis. Osmosis, simply put, as I mentioned, means diffusion of water. Now, as you can see in this picture, water can diffuse through the plasma membrane by itself without the help of any other molecule because it is small enough to seep in and out of the plasma membrane. Or it can use channel proteins you can see in purple and are referred to as aquaporins to move across the plasma membrane. So osmosis, as I mentioned, it simply means diffusion of water using either the plasma membrane directly or passing through the plasma membrane directly or passing through the channels that are continuously open. Here's a scenario that describes basically the 4 different types or 3 different types of diffusion. Simple diffusion, passing through the plasma membrane. Carrier-mediated diffusion, where proteins pass through and it causes the change in the shape of the carrier protein. Channel protein, which we broke down to voltage-gated channels, chemically sensitive voltage-gated channels, and leakage channels. And lastly, osmosis, which is movement of water with its concentration gradient. Now moving on and talking about osmosis and diffusion, the next word that comes up is the word osmolarity. Osmolarity is a way to measure the number of particles within a solution. Number of particles or number of solute particles in the solution can be measured in osmolarity. It is different from concentration, which is measured in molarity, and is typically used in chemistry. One thing you need to understand is this, that when you have a solution, if you have a lot of solute molecules in them, or you have high osmolarity basically, that implies that you have very limited water. And if you have a dilute sample, when you have a lot of water, in relation to the solute molecule, your osmolarity would be lower. So, osmolarity is low when your sample is dilute, and osmolarity is high when your sample is concentrated. Now, keeping that in mind, we are going to focus on a concept, and I'll come back to the couple of slides. We're going to focus on a concept called osmotic pressure. Osmotic pressure simply means movement of molecules, pull solute molecules have on water. Pull solute molecules have on water. Simply put, is if you have a situation where you have more solutes, that solute is going to attract more water molecules toward them. The example I can provide for you is a scenario where you eat a bag of chips or you eat a lot of sweets. In either of these scenarios, one of the first things your body requires after eating salty or sweet material or food is basically drinking water. If you think about this, you can bring it down to a much smaller level at the cellular level, and you can draw the same conclusion, which basically means if you have a lot of solute inside the cell, those solutes are going to pull water into the cell. And if you have a lot of solutes around the cell, those solute molecules are going to pull water out of the cell. So keep that in mind, and we'll come back to this in a minute. So let's keep that in mind and a concept of diffusion and osmosis and apply it to these two scenarios that we're going to discuss. In the first scenario, we're going to start with two different solutions that are separated by a plasma membrane or a membrane, which you see as these yellow dashes. This this membrane is not selective, rather it is permeable to both the solute molecules, which are the red dots, as well as the water that is represented in blue color. So basically what that means is, if you have a high solute concentration on one side and low concentration of solute on the other side, what both of these molecules are going to do is they're going to basically use the process of diffusion and osmosis to reach equilibrium. So in this case, you can see that solute molecules are high here and low in this side. So solute molecules are going to diffuse across the plasma membrane and come to the left-hand side. And water, on the other hand, is high here, so it's going to move on the opposite side. This movement continues to happen until you reach equilibrium for both water and solutes on either side of the membrane. And you can see it in this side. So equal distribution of both solutes as well as water molecules on either side. Now, this is a scenario where you have a permeable membrane to both solutes and water. However, in the second scenario, your membrane has become selectively permeable, as you see the yellow dashed lines. So here you have same situation, lots of solute molecules on this side, a little bit on this side, and therefore in relationship you have relatively more water compared to solutes on the left-hand side. Now in this scenario, you cannot move your solute molecules via the means of diffusion to the left-hand side because your membrane is not permeable to solutes. But what you can do is you can move water to basically via the means of osmotic pressure. Remember, osmotic pressure, as I mentioned earlier, is the pull solute molecules have on water. And the more solute you have, the more water is going to be pulled in that same direction. So because you have more solute molecules on this side, water is going to be attracted or pulled toward that side. And this is the scenario you will get. Higher concentration of water on the right-hand side and lower concentration of water on the other side. Again, notice that the solutes have not moved in this scenario, but now you have a level of equilibrium between water and the number of solute molecules that are present on either side of the membrane. Now, going back to the type of pressures that are involved in the cell, we already talked about osmotic pressure, which again, basically extremely important. Pull solute molecules have on water. The more solutes you have, the stronger the pull. And the second pressure is hydrostatic pressure, which is basically the pressure the water exerts on the plasma membrane. Now let me clarify this because there is a problem with this, the description that's written here. So it says pressure of water inside the cell pushing on the membrane, but the reality is this. If this is your cell, you have water inside the cell and you have water outside the cell. So you can have hydrostatic pressure pushing in, hydrostatic pressure, you can push in the membrane trying to get out, and you can also have hydrostatic pressure pushing back on the membrane from the fluid that surrounds the cell. So you will still have hydrostatic pressure here as well. So it's not only pushing on the membrane from inside toward the outside, but you also have hydrostatic pressure moving from outside into the inside. Now I'm going to stop the PowerPoint here and we'll pick up talking about tenacity in the next section of the PowerPoint.